For the organization. It's also nice that for traditional reasons the CNS also always has a place in this symposium, although the news that we have in the brain tumor field are either largely disappointing or even bizarre. And I'm going to show both of that with you. So I think it's important to know to start that immuno-oncology is really not important for most of the diseases we treat. So when we talk about CNS and immuno-oncology, we usually think about glioblastoma, but glioblastoma is just about 15% of all our tumors. So for most of our tumors, good old traditional neurosurgery is still good enough and provides a cure. And we are really talking now about a minority of primary brain tumors. There are still, after decades of very intensive research, some important questions that have not been really addressed for glioblastoma. First, we don't know where the tumor comes from. So if we think about profiling, looking for antigens, we really would like to understand where the cells arise. We have various types of brain cells. We know that glioblastoma cells can be induced to differentiate in almost anything that you find in the brain, but we have a very poor understanding of what the cell of origin is until today. If we believe it's a stem cell, then we also have to account for this observation here, that is that the lifetime risk increases as you go into age, and probably if we would all get old enough, maybe this would really be a prevalent disease in the end, a very important disease, like lung cancer. It's not at the moment, but you see that the increase is really very steady. And then if you take into considerations that most people of my age probably don't have any stem cells, any dividing cells anymore in the brain, the question really is, how can we generate tumors in a post-mitotic tissue? So that is another open question. One hypothesis that is almost supported by nothing, but is very interesting, nevertheless, is that this increase is a result of immunosenescence, that the immune system is not controlling anymore what's going on in the brain. As I say, very attractive, but unfortunately not backed by a lot of data. We have 5 fields of interest at the moment that you may want to subsummarize under immuno-oncology. We can argue whether ADCs are immuno-oncology. Many people think it's not, but I also have to show you something positive, so I include it. And then you may want to argue whether oncolytic viruses belong into immuno-oncology, and here I think the evidence, at least in the brain tumor field, is increasing that oncolytic viruses, if they work through the immune system. So I will start with a bit of positive news about a new antibody drug conjugate that is in last steps of clinical development. It is based on targeting EGFR, which is still one of the most prevalent and probably most important molecular lesions in glioblastoma. If you ask simply for overexpression, it's up to 60% of all patients that have overexpression of EGFR. About 40% of all patients have amplification of the gene to various levels. And then about half of these patients with the amplification have the specific mutation referred to as V3, which has been a target for vaccination. And we've heard this morning from Dr. Bargou that CAR-T cell approaches against V3 are underway. And I'm going to share with you some data in the end showing that that is probably not a good idea because it's probably not going to work if you target this antigen. But the essence here is that EGFR, if it is active, changes its conformation. And that has been used initially to develop an antibody called A06 that is only able to recognize EGFR if it is in this conformation. That shows the binding site. Now, this antibody was interesting for immunochemistry of flow cytometry, but it had no intrinsic activity. So tumor cells don't care if you treat them with the antibody. But if you link the antibody to this toxin, MMF, then you have an ADC that is relatively active, at least in animal models. Now, it is interesting that we are not really advancing our understanding of how the EGFR amplification translates into that confirmation. And the critical question that cannot be addressed is this one here. So this is the inactive variant. You need to open it up for the antibody to bind, whether it's the native antibody or the toxin. And it's not really clear who is triggering the unfolding, because the ligand can only bind the molecule if it unfolds. And it's not really clear what comes first. Does the ligand first see the molecule and then it unfolds? So this is something that we try to understand. Also, many companies, I think, are working on that because it's important for this overall concept. In any case, what is important is that this V3 or delta 2 to 7 mutant, which has a deletion up here, only exists in the unfolded form, like that, and that's why this antibody will always bind to cells expressing that variant. The EOTC has done a trial with AbbVie. It's the 1410 trial of the EOTC, which has bizarre design in a certain way, and the bizarre design is based on an animal model, U87MG, that most people think is completely irrelevant. But in retrospect, it was a good idea that the trial was done that way, although we really argued against doing it this way. So patients had their tumor tissue tested for EGFR amplification, and they were enrolled at first recurrence to receive the ADC alone in combination with temozolomide, because that combination worked so well in the irrelevant animal model. And the control arm is always a debate in glioblastomas. The control arm is complex. The control arm is temozolomide re-challenge one more time if the recurrence is relatively late, or lomustine, another alkylator, if the recurrence is relatively early. And that, of course, produces a lot of bias and very many small subgroups. So that may not have been a good idea. In any case, that's the only data I want to share with you today. It's unpublished, but it has been shown at meetings. So this is only the comparison of ABT414 plus temozolomide against the control arm. And after a lot of very, very frustrating curves, where you really could save the money for biometric analysis because it's completely negative, we have finally a trial that looks positive. It is formally not positive because the OTC did the analysis too early, so we were just scratching significance. Now with more follow-up, it looks significant. There are a lot of questions. What we do not understand is why this antibody-drug conjugate doesn't work alone. It works only in the combination with temozolomide, and that's very difficult to understand. And there has been a lot of arguments what to do with these data, but anyway, the company, and I think it was a good idea, has decided early on to go into the first-line setting, and this trial is already fully recruited, so we have data that are maturing in the first-line setting, but of all drugs that are around at the moment, that drug may be the next one that is going to go for approval in glioblastoma. Now I switch to the real immuno-oncology. And most of the immuno-oncology approaches in glioblastoma are based around the fact that we think that immunosuppression is really a key feature of these tumors. A lot of research is focusing on the so-called bad macrophages and monocytes, which are studied in various disease entities. In glioblastoma, it's a very, very prevalent type of cell because we have the microglial cells around, and there are glioblastomas that have more than 20% microglial contamination. So it's really an important population. We have a lot of soluble mediators that are immunosuppressive, and we have the classical tumor cell, immune cell interactions that are being discussed in other cancers as well. In terms of the general efforts of targeting the immunosuppressive mechanisms, we have one key area of interest, which is obviously checkpoint inhibition as anywhere else. Specifically in glioblastoma, we have some phase 2 randomized data on TGF-beta inhibitors that all have been not successful. Probably not because the target is wrong, but because the drugs were either not working, not reaching the target, or too toxic. So I think as a target, this is not dead, but we need to do better. And there is interesting data upcoming in other cancers in the last weeks in Nature of combining checkpoint inhibition and TGF-beta inhibitors, and that's what we're also doing in the lab at the moment. There's a lot of interest in tryptophan metabolism in glioblastoma, and actually this was one of the first cancers where that has been studied, but no clinical data yet. And there are developments of STAT3 inhibitors because at least some groups in the US think that a lot of the immunosuppression is STAT3 mediated. So in terms of checkpoint, just to summarize the preclinical data that we have at the moment, there certainly is ligand expression in glioblastoma tissue. Whether it's all on the tumor cells or on some of the immune cells that are invading is not clear. It's not clear, at least in brain cancer, whether the level of expression matters and whether this is a predictive factor. But in any case, before something can be called predictive, we need to show that something works at all. So that is the major challenge at the moment. If you look at the public databases like TCGA, you cannot derive convincing evidence that a glioblastoma that has a lot of PD-L1 is worse than any other glioblastoma. And there's a lot of promising studies in animal models, which is not surprising because our animal models are somewhat artificial. They have a high tumor mutational burden, and that's why they probably respond well to checkpoint inhibition. I would like to share with you the data from the only completed and mature data on checkpoint inhibition in glioblastoma. It's still not published, but it will be published soon. This is checkmate 143. I think everything about this trial is okay, good quality, except the hazard ratio in the end. So it's one of the few checkmate trials that is not positive. This is a trial that is dominated by US standards of care, and that's why bevacizumab is the control arm in recurrent disease, which would not be an appropriate control arm in Europe because it is not approved, but it's primarily, or it was primarily, a US trial. So patients were enrolled at first recurrence after standard of care. It's a relatively large trial. Patient characteristics are relatively unremarkable, except maybe what is interesting is the biomarker approach. So it was predetermined that a tumor that had more than 1% PD-L1 expression would be considered positive, which is an interesting benchmark, 1%, because I think it's unlikely that if target expression is important, it would be sufficient on 2%, but that would have been a positive tumor. And interestingly, according to their test, and many of you know that there's a lot of debate on how to best assess PD-L1 expression, but in any case, according to this cutoff, their method of assessment, only a quarter of glioblastomas have more than 1% of PD-L1, which predicts probably that the treatment cannot work if that would be important. That doesn't make a lot of sense. The other thing that is important in glioblastoma always is looking at steroids, because obviously patients on steroids may not be the best patients for an immunotherapy trial. The cutoff that was chosen was 4 milligram, and you see that a minority of patients were on steroids when put on the trial, but that was included as one of the subgroup analysis. If you look then at treatment exposure, then treatment exposure is, of course, predicting the outcome. Patients were a median of 2.3 months on treatment. And if you consider what we feel about how immunotherapy should work, it is probably clear that 2.3 months is a very short duration for immunotherapy to work. And patients were almost double as long on the standard of care arm, which was bevacizumab. This translates into a progression-free survival curve that shows a clear inferiority of the experimental arm. And what you can also see is that most patients drop off the trial when they have their MRI scans, right? That's why the curves look like that. This is always when patients come to the MRI, they are diagnosed to be progressive. They're not clinically progressive, they are radiologically progressive. And that happens roughly 1 to 2 MRI scans later if you are on bevacizumab. And then if you look at overall survival, the curves are kissing and crossing several times. That is clearly a negative trial. Now, of course, we had to digest these disappointing results, and then everybody does what shouldn't be done. You go for subgroup analysis. And then my personal interpretation would be that probably all these subgroup analyses are noise. But if you have a lot of fantasy, you could still look at PD-L1 expression. And then you know how these curves look, right? So if a dot goes to the right, it favors bevacizumab. If it goes to the left, it favors nivolumab. So if anything, having PD-L1 is not good, which doesn't make any sense, but that's what it is. And then you can also look at dexamethasone, and that may be a little bit more interesting. Again, very, very small effects. If you are not on dexamethasone, you may have a minute benefit. And what is more logical maybe, if you are on dexamethasone, then you probably have a bigger tumor if the oncologists take the right decisions. And if you have a bigger tumor, you may have more benefit from bevacizumab. So that makes a little bit of sense. But all these effects are really minute. So what do we do with this? Usually these days, the important decisions are always made early on. So the decisions to move into first line were taken long because these data were available. So based on MGMT status, which is still very important in the glioblastoma field, we have two trials, one for MGMT unmethylated patients. And in this scenario, the very self-confident rationale that I certainly shared is that in unmethylated patients, temozolomide doesn't work anyway, so we drop it in the experimental arm and just have to show that nivolumab is better than something that probably doesn't work. This trial is fully enrolled. The data should be in more or less, and we don't know what the data is, and that sometimes is not such a good sign. And this trial here, the methylated trial, was initially started as a phase 2 trial, but then based on the data that I have shown you before and a possible hint that patients who are MGMT promoter methylated stay longer on treatment and may have more mutational burden because you hit them constantly with alkylators, this has now been turned into a phase 3, also full independent phase 3 of its own, and this is still enrolling, but probably only for a few weeks. So this is where we stand with checkpoint inhibition, and all the other companies are far behind, and the activities with pembrolizumab have been sort of slowed down quite a bit after the results of checkpoint 143. Then we have several approaches that target glioblastoma specifically, and a lot of these are vaccination approaches. And we have 3 phase 3 programs that are summarized here that I'm not going to cover in any detail anymore. The first one was very simple. This is just a vaccine with one peptide targeting a mutation that is specific to the tumor, should be completely untoxic. The second one is more complicated. This was a dendritic cell-based vaccine, again taking off-the-shelf 6 peptides, same size for all patients without any tumor-specific adaptation, but much more laborious because it required the dendritic cell preparation. And the third one, DCVax, is the most complex and the most difficult to predict because this is a vaccine made of lysate from each individual tumor, so each patient has its own vaccine developed. So this is more complex. And the results are altogether hurting. So I would say in summary, we have 4 big efforts that have failed for various reasons. This one I have shown you, this is the nivolumab trial. The first one is the vaccination trial against the V3 mutation. This trial, of course I'm biased, but I would say this was a very good trial. Everything was done well. It was done in cooperation with the EOTC. It's just negative. It's bluntly negative, so I don't show anything in terms of the data. After this trial was negative, the second trial that we also did within EOTC was terminated because the investors pulled out of this whole program because apparently it was not possible to raise money anymore. And then we have the third trial that has now been published recently. This is DCVax. And I'm not showing the data because I think the data shouldn't be shown because they are bizarre. And also, of course, if you see this is a phase 3 trial, a randomized trial, and if you see that this was published in the Journal of Translational Medicine, then you wonder whether these authors are very modest, but this is not usually where you send these papers, right? What they have done in this trial is they have hidden the primary endpoint of progression-free survival, and they just published the overall survival pooled from both cohorts. I think this is a quite unique publication strategy. So you don't really know whether the treatment has done anything. You just know that if you were enrolled into this trial, you lived relatively long, which is something that we often see in Immuno-Oncology these days, that patients put onto clinical trials live much longer than patients outside clinical trials, and I think that's a general experience in oncology, but it's quite striking. In any case, I have then taken the exercise to do the same game with our own trial, and if we do the same thing, we also pool control group and experimental treatment all together. It's roughly the same data. So there are two options. Either we will never see the whole data, or we will see the data in some years, and then we can see whether it's going to be positive. But if you're interested in bizarre publication strategies, look at the DCVax publication. So where do we stand now? And we have a lot of interesting targets. There's a lot of interesting preclinical work ongoing, but I think in 2018 it's fair to say that our field has been shaken a little bit, and I think it's certainly not time for big phase 3 trials at the moment. I think we need to go back to better science-driven trials, and I don't think that more one-size-fits-all trials in glioblastoma are going to work. We have some interesting observations. We also have our little CAR field in Immuno-Oncology. So the first studies were obviously simply feasibility, the question of whether you can safely inject CARs into the brain. That appears to be feasible, but it's probably a question of the dose. And then there was this one paper that we all loved because it looks like a case report. It's a very interesting case report of CAR T cells interestingly injected into the CSF. I would never have thought that that can work. I would always have thought that if you put something into the CSF, it will still stay there and maybe cause a bit of inflammation, but nothing else. But this patient experienced a regression, at least for some months. That looks convincing. It's actually not a case report because several other patients were reported in the same paper and they did not respond, but at least something that works. Now, the concept of CAR T cells has been mentioned before. I'm not going to show it in detail. It's, of course, very interesting. It will also be challenging. It will be expensive. But the key problem, again, for us is this little question here. What do we choose as a target? And I think this will be the decisive question for glioblastoma. We have no targets. The one target that we thought we had is the EGFR V3. I've not shown you the data. You know, the vaccination trial is completely negative, and one reason why the vaccination doesn't work is that this antigen is extremely unstable. And there is, again, a very small paper in Science Translational Medicine where CAR T cells against V3 have been used, and the main observation was that very shortly after these CAR T cells were applied, the V3 antigen was lost. So the cells are not stupid. They simply downregulate the V3, and then you can forget about these T cells. So I think CAR T cells against V3 is nothing that I would put any money on at the moment, and we have no other good, convincing candidates. Now, there are, of course, other approaches. You can be a little bit more courageous and say, well, I need to move an efficient immune response into the brain. And something that has been developed, I think, also in the hematology field and that we have now adopted is using the mechanism of danger signals like MIG-A or ULBP, which are expressed on tumor cells and virally infected cells. And these signals trigger an interaction via NKG2D on NK and T cells. So everything that looks strange, bizarre, expresses these danger signals, and then the immune system can potentially attack that. This sounds like this approach would have a lot of potential for side effects, but this is not what has been seen in the first phase 1 experiments in humans that have been done. And we have always been quite interested in danger ligand expressing on glioma cells, and we have some nice data generated by Tobias Weiss and Patrick Roth, who many of you know. So we have been working with CAR T cells in a mouse syngeneic system that have as a mode of action the NKG2D moiety as the recognition signal. So they will interact with any cell in the organism that expresses danger ligands, and at least in some of these mice, they are very small numbers because it's a very complex type of strategy. This seems to work, and we are negotiating with a company that is working on these cells to see whether we can get a clinical trial, first clinical trial in glioblastoma on the way. Now I would like to conclude with the oncolytic viruses. And there has been a lot of discussion about polio in the recent 1 or 2 years. And last year, I think it was when the Cancer Center had the Patient Academy. I also talked a little bit about that. And the day before, I went to the internet, put in polio and glioblastoma, and I got these beautiful pictures. This woman has become very popular in the US. So a lot of people have been talking about polio and about the experience made in Duke. And I think most of the community has been sort of very critical or ignorant, or we didn't really believe that this is moving forward. And anyway, it's coming from Duke, and who believes data from Duke. And then finally, the colleagues from Duke found 2 or 3 referees that believed in the data. And these referees were working for the New England Journal. And so this is a few weeks ago, 3 or 4 weeks ago. And this is really, again, it's an interesting publication because I think most of us would never have dared to send that to the New England Journal, but maybe you should be more courageous. So don't think that's a randomized trial, right? This is, again, something that should be forbidden everywhere in the world. So the blue is the patients treated with the poliovirus. The red is a control group that they constructed. And of course, you should not put that into the same graph. And we all know that, but apparently in the New England Journal, you can still do that. And then, I don't know whether you can read that well. You see, so this is all recurrent glioblastoma patients, and even with their control group that was probably selected in a somewhat biased way, you see patients just die. And they die until 18 months, and then they have this blue curve, which is not really a curve. This is their so-called long-term survivors. And you see, if you can see that here, this patient is censored, this one is censored, this one is censored. So you have a lot of censored patients here. So most of the survivors are censored. We are talking about, here we are at 6 patients at risk who remain. It's non-randomized, it's actually non-controlled. And you can see if you go into the supplement, you find that 1 or 2 of them are IDH mutant patients, so they're not really glioblastomas. In any case, now it's out there, and the pressure increases. And it seems that the FDA is interested in giving accelerated approval based on these 6 or 7 patients. And our American colleagues think it's unethical to do a randomized trial because it's so good. And the Europeans, EOTC, we are very reluctant to even engage in discussions on single-arm trials. But this is the scenario now. This is the data, so if somebody asks about polio, you know what this is based on. And they are reasonable scientists who have been doing it, and they are absolutely convinced. I know many of them. What can you do? They have a nice paper. So with that, I would like to conclude. I think that, yeah, I like the research area, otherwise I wouldn't be there. Not everything is completely negative, but a lot. And of course, like many other disease areas, I think we need to be clever. We need to combine, and we need to base our treatments on some real science and biomarkers and not just treat everybody the same way. Thank you very much. So I'm also the moderator. Does anybody want to ask the speaker a question? Otherwise, I'm also there in the coffee break. Yeah.
So I agree with you 100% about your comments about the poliovirus study. These data actually have been presented in oncolytic virotherapy meetings for at least the last decade since I first saw one of their purported complete remissions. And so it's a very interesting observation that they could get these data published there. But there actually are much more, I think, robust data that you didn't mention, presumably because of lack of time, from Nori Kasahara's replication-competent retrovirus approach. Do you have any thoughts about that, or is that not something that you think also has legs?
Well, there's no controlled data in the public domain, right? And actually, I mean, you're right, these sort of tails have been reported with a lot of oncolytic viruses. Also, what Nino Kioka is doing at MGH, he also says he has a tail. And actually, Nino Kioka has written now a big review in Immuno-Oncology collecting all the phase 2 trials in recurrence that have a tail. With the virus you mentioned, I'm not so sure how good the data is, whether that this is related to immuno-oncology, but the same is true for the polio. We also don't know whether that is related to immuno-oncology. Okay, so I think you can continue.